Good morning. Let's start our day. I would like to introduce Professor Timothy Zhu at University of Pennsylvania. Thank you, Professor.
Thank you very much. It's early in the morning, so I hope everyone, thank you for all coming so early. So what I'm going to talk about today is a little different from before. We're more concentrating on clinic application and dosimetry, particularly for pleural treatment. And let me go from here. I'm going to go back to look at the background components of PDT and talk about what is needed for explicit dosimetry. And then we will go over specifics for pleural PDT. And I'll talk about a navigation system. In the end, I spent some time to describe a macroscopic model, which is actually a calculation-based model. And it's sort of taking the second approach, as Dr. Wilson has mentioned before yesterday. Very nice summary. And it's an indirect method. And talk about that in some detail, and also in the end talk about the future directions. So as you are well know now, the PDT really depending on three components: photosensitizer, light, and oxygenation. As a result, all three components is needed. And here is just a brief overview of the mechanism of actions. So patient is giving photosensitizers, and then what activated with light. The activated sensitizer, most of them is going through a so-called type 2 interactions, which directly interact with oxygen to produce singlet oxygen. And that actually is the pathway for singlet oxygen-based modeling. And that's also the pathway for the majority of photosensitizers that's clinically available for the trials. And there's also a possible type 1 interaction where, free radicals other than singlet oxygen is generated. And in the end, they all do the cytotoxic damage through this cytotoxic oxy product, doing the cellular damage. So really, there is two things to target the tumor. One is through the sensitizer, and the other, of course, is using the light as a control element. And sensitizer, I talk about that briefly, usually is delivered in vivo prior to surgery or treatment. They're supposed to have a higher concentration in patient, retaining tumors compared to normal tissue. And toxicity mainly is sunburns for the first generation sensitizers. So photofrin is the one that's first generation, partially purified porphyrin monomers. Activated at the 630 with 5 peaks, and the main drawback is the lasting photosensitivity and for 4 weeks, and also the skin toxicities. So here is the 5 peaks, and we are treating usually at 630 because the light penetrates deeper into tissue at the longer wavelengths. But all the other wavelengths can also be used even for treatment too. Second generation photosensitizers commonly has a longer wavelengths and also more purified. So I've listed here just a few that's already is in the clinic trial stage. 4 scan, as you all well know, has been extensively used, approved in Europe for FDA approved for PDT treatment. Vodiporphyrin is also FDA approved in US, at least for AMD treatment, and also have a lot of clinic trial ongoing for various diseases, including prostate, for example. It's going to come up. ALA and the HPPH, that's the one that's developed in Roswell Park, Lutex and TUCAT. So the ones I mentioned a little bit more here is the HPPH. That's the one drug we're currently using for pleural PDT. And it has pretty clean absorption peaks, has 2 peaks, one at the near 400 nanometer perfect for excitation with blue light, and the other at 665 nanometer. And the fluorescence peak is a single peak, which is reasonably strong at a wavelength that is a bit overlapped with hemoglobin absorptions, but still is a clear peak. And typically, absorption spectrum is the one that one can use to determine the sensitizer concentration. And the other important thing of using the absorption spectrum is one can use that to determine the optic properties, and that is important for the calculation of light propagations. And the third one, of course, is the determination of oxygenation by exploring the difference between oxy and deoxyhemoglobin. And a contact probe, you probably have seen again and again, basically is a probe you can make with a single white source detector, multiple detector sources coupled into a monochromator and CCD camera, and that can be used to resolve such as optic properties. So here is one example. There you measure data at known distance from the light source. We'll be able to fit into a semi-infinite model, which the model was pioneered at Brian Wilson's group and Mike Patterson's. So there is well-established formulas that you can use to fit those measured data in the semi-infinite mediums, and that will be the fitting will let us to determine the absorption and scattering coefficient of the tissue. For example, you can do the calculation and determine the penetration depths of light at the treatment wavelengths. This is at 730 at different for different organs. So you can see there is a large variation of optic properties of penetration depths depending on the tissue types. So for those more complicated treatment with various different tissue types, if we want to know the penetration of light into the tissue, this quantification is very useful because you can see it changes from up to 5 millimeter in skin to as short as less than 1 millimeter or 1 millimeter. It's in liver, it's much shorter than the other organs because of the hemoglobin, higher hemoglobin concentration. And here is again a diagram that shows the absorption spectrums for oxy-deoxyhemoglobin, and there is a very large difference of the two between 600 and 700, and that can be easily used to determine the oxygenations in the tissue. And here is some data analysis use the component method that can be used to determine the oxygen saturation in the tissue for different organs. This is done by postdoc Wang and in Arjun's group and together with us for IPPDT, and that was done published in 2005. And here is the result for various different tumor, small bowel, large bowel, peritoneums. And you can see some kind of a distribution for oxygen saturations and total hemoglobin concentrations, and also a very various distribution of the drug uptake. In this case, photofrin is used. And the same method can be used to look at the spatial distributions of these various quantities. This is a data taken in a prostate samples in vivo in a patient, and you can see actually there is a large change of total hemoglobin concentration, not much change of oxygen saturation or the drug concentration. I mean, there's also a big change of drug concentration too. So these, variations could be related to different malignancy tumors or whatever inside the tissues. And the absorption method when used can be used to determine the distribution of drug concentrations. And here is a Lutec concentration in nanogram per milligram or kilogram per microgram per kilogram, same unit. And you can see it changes by a factor of 5, almost 4 times in the same patient at different location of the patient. So if we believe a PDT dose is important quantity for dose for the PDT effect, then one really has to deliver more light in the region where there's less concentration of sensitizers than when there's a more concentrated sensitizer. So these data strongly indicates there is a very heterogeneous optic property distribution as well as sensitizer distributions in patients, and that really strongly indicate one has to do a personalized dosimetry, patient-to-patient specific dosimetry. So the next one is light. So we have mentioned in my last lectures that there's 2 quantities, irradiance and the light fluence rate. Fluence rate is accounting for all the light into these small volumes divided by this area from any directions without regarding to where they are coming from. And irradiance is usually is only accounting for normal incidence. So if you're shooting at an angle, then your irradiance value will be reduced by a cosine theta. And really for PDT, this is the most important quantity. But irradiance can also be often used because they can nicely correlate with the calculation. If you know the power of the light source in a simple collimated light geometry, irradiance can be simply calculated by the treatment area and the power of the light at the location of treatment and divide by 2, right? But there is basically a difference between these 2 quantities, and this one is typically larger because it takes into account the scattered light. And one can measure either one with either a flat detector. This detector measures irradiance. So these are the detectors we used in the early days for the pleural treatment, where you can suture that detector directly into treatment interest locations. And nowadays we're using this isotropic detector which measures the light fluence rate. Dosimetry system, you can measure that in multiple sites. So if one has to compare if there is any difference between these 2 methods of measuring light. We have find out there's a great deal of heterogeneity in light dose. So if one uses the irradiance measurement, you are not really taking into account the heterogeneity of the tissue optic properties because all the scattered light is not being counted. So one actually would want to use the light fluence rate measurement directly, which will account for the patient-to-patient difference due to the tissue heterogeneity. This is again a figure that shows if you look at the ratio of isotropic detector versus flat detectors, and it is actually at the tissue surface. It's a function of optic properties. Here is plotted against the effective attenuation coefficient versus a different scattering coefficient, for example. So they actually can be described very nicely with diffusion series. So this can be calculated analytically very quickly, and measurement and the calculation agrees reasonably with each other. Here is the result for the pleural PDT treatment in the patient using our current method of light delivery, which actually is using a point source moving around inside the pleural cavity. We looked at the 12 patients. So what we're basically doing is just suture the light detectors and isotropic detector and flat detector side by side. And then you just accumulate during the entire treatment, and then you acquire data from both detectors, and then you can plot out this curve and then fit it to a linear curve. You do that for various sites. Here I am showing in one site, that's apex, but we can do it in all the sites we use. Typically we're putting 7 detectors, so we actually do the comparison in every single site. And then determine average ratios, 1.7 for example in this case. And we have done that for, again, 12 patients, different sites, apex, posterior sulcus, posterior chest wall, anterior sulcus, anterior chest wall, pericardium, that's the heart, and posterior mediastinum. So, and all these can be used to determine the so-called K coefficient, that's the ratio between the two. and the standard variation, and that can be used to determine this standard ratio. So this actually is the number we use currently to convert the flat detector reading to our current dosimetry reading using isotropic detector. And the last one, of course, is oxygen. And so certainly PDT is really completely oxygen dependent by its very definition. And without presence of oxygen, there is no therapeutic effect. And the method we're currently using, it is really a difficult problem to in vivo determine the concentration of oxygenation in tissue. So we can use the so-called oxy and deoxyhemoglobin concentration using the contact probe measurement, and that can be used to, doing some calculation to get some calculated value for hemoglobin concentration. No, not oxygen concentration in the tissue. So I'm gonna switch to talk about the PDT for lung cancers. We have two trials going on. One is a phase 2 trial for HPPH, which uses 2 milligrams per kilo of photofluorine intravenously given to the patient, 24 hours, 630 nanometer light delivery, up to a dose now is 60 joule per centimeter square. That's a light fluence rate measured in vivo using the dosimetry system. This particular, disease really is very late stage disease. So the prognosis is very poor. The patient don't expect to live more than 6 to 18 months, even with surgery. Surgery is the conventional treatment. So we have done that for a long time, actually. And if you look at the most recent analysis, which is analyzed the patient from 2004 to 2008, you can find actually the median survival for this particular mesoradical prostatectomy with PDT is very long. This is 4 years out, still is, a long, for a subset of patients. So there is actually a difference between a patient with epithelium prostatectomy and radical prostatectomy, and that's the way actually we have been using for all the patients, which basically is preserve the lung during the surgery rather than cut the lungs out. So we actually have very good outcome data, and hopefully, the PDT can be used to, further improve this disease treatment. And in terms of treatment, we put in 7 detectors in various locations in the pleural cavity, and the goal of the treatment is basically to cover the entire pleural cavity with PDT treatment to kill the cell up to about 5 millimeters in the entire pleural cavity. And after the tumor is removed. So one of the criteria is that the tumor has to be completely removed to a residual tumor of less than 5 millimeters in depth. Isotropic detector, this you can actually commercially buy it from MedLife, for example. This is probably one of the only commercial companies. You suture these detectors into various locations using sterile tubes, and the detector itself actually doesn't need to be sterilized. We don't sterilize detector because it's inside a protected tube. And the treatment is kind of complicated in that you have to, the current method is using a light source, and you move it around inside the cavity to treat to various locations. And so there's 7 detectors in, so you basically know where the light dose is in these 7 locations correctly. Of course, there's lots of complication because you can see there's a big cavity here. So there is lots of light, internal scattering, and light bouncing back to each other, as well as absorptions due to bleeding, for example. And typically, the surgeon is doing a very good job to stop all the bleedings, and you pour in intralipid into it to dilute the blood too. But sometimes you can see bleeding in the cavity, so you have to pump out the blood and put in clear diluted intralipid to make sure it's not absorbing all the lights to reach the surface. And it's a very long treatment, typically an hour to up to 2 hours treatment, depending on the cavity size, because it's patient to patient different. So we have been doing this light dosimetry for a long time. Then the next question is, of course, how do you guarantee the light is treated, homogeneously all the way around in the entire pleural surface? Our current method is poor man's way. It's the surgeon decide how they treat it, because they basically use one detector to treat the entire area by moving it more uniformly in other areas. So it's really, depending person to person dependent, very skill dependent treatment method. And if you assume the scatter light dominates, it's actually one nice thing in an integrating sphere is if you know the light source power, you can calculate the scatter light, which is a constant in a perfect integrating sphere, and that can be calculated in a sphere, for example, by the power divided by the surface area of the sphere, and multiply the diffuse reflectance. And that, again, diffuse reflectance, of course, is tissue dependent. So you have to know the tissue optic property, and there's a well-established formula to calculate diffuse reflectance once you know the tissue optic properties, and that will allow you to predict the light fluence rate. And one thing we are doing now, since we have accumulated large amount of data for light fluence versus the volume and the treatment time, so we sort of try to see if we can correlate the treatment time to the surface area of the cavity, because we have a large collection of patients that have been treated. So based on this model, if the scatter light is the dominant factor, then they are proportional to each other. And if we are thinking if it's an integrating sphere, perfect integrating sphere, then the shape of the integrating sphere doesn't matter. So it can be any shape, right? No. So that just important thing is determine the surface area. So we can calculate the K value based on the previous formula, which I have shown before. If you use this formula and flip it over, basically, you know the light fluence rate is equal to the total treatment times, is equal to the total treatment fluence, which we know, divided by the treatment time, okay? So the treatment time is a function of total fluence, which we know, and we are changing somewhat for different patients, but it's a 60 joule per centimeter square, and surface area, and the optic property is one thing we actually happen to measure for each patient. We have measurement at least a few points for all different type of organs. And the surface area is very complicated. You can calculate that by we currently using the prior CT data. So you can see that they change quite a bit, and then you can calculate the volume based on the lung cavity or the surface area from this pre-treatment CTs. And then the time is known, the volume can be determined, and there's in the early days actually we have some of the patient that the lung is preserved, so you have cases where the lung is still inside, and most of cases, of course, the lung, actually in the early day we have lung taking out. Some cases, nowadays actually lung is always inside. So we can actually correlate the relationship between the treatment time and area, finding out the mean slope. And we also can calculate the diffuse reflectance. And as you can see, actually it's going all over the place, starting from 20% all the way up to close to 80%, depending on the patient. This is the data measured for all the patients put together. So these data is a bit messy, quite messy actually, but so this give us a starting point to how to calculate the light fluence rate. So we actually want to really determine that very rigorously. So what we are doing now is to introduce a so-called navigation system. The way we do it now is we have infrared camera that can be used to track the movement of the laser source in real time. And so we actually have another so-called one, so that it's infrared camera which can detect the position of, say, 4 refractive markers. And from these 4 refractive markers, you can determine the position of this tip to an accuracy of sub-millimeter, very accurate. And in a very real 20 frames a second, real-time speed. So we can use this device to determine the treatment contour now. So we actually can determine the surface contour of the treatment area. And if in this particular case, you can also see where the detector is, we can use these ones to touch on the detector, decide where we put our detector in the treatment area. And here, the red part actually is the tracking of the laser source during the entire treatment procedure. So we can track where the laser is. This is what it looks like, and the camera here looking over. This is doing the treatment. There's a this is actually custom-made refractors which allow one can see larger angles rather than within 90 degrees. So you move it around. And the simplest model is to assuming you have a direct light, which is very easy to calculate. All we need to know is the source power and divide by 4 pi r square. R square is the distance between the source itself, we know perfectly well, and the surface area where we want to treat, where we do the calculation. Certainly, scatter light is important. As I mentioned before, our previous hypothesis of data analysis actually is scatter light is dominant. When we do this calculation, we started with direct light. And actually, we have some phantom study that shows the scatter light is important. But if one want to find out the fine features during the treatment, direct light will become important too, because all these fine features actually is dominated by the direct light from the light source, especially when the light source is really, really close to the tissue surface. So these 2 components is all accounted for. So here is the comparison at various sites between the calculation and measurement. Here is just looking at direct light itself. And here is the measurement. So blue is the measurement and for various sites. Typically, the calculated direct light is always less than the measurement. And our explanation is because of the scatter light. And the difference actually, we currently are modeling it as this difference is a constant fluence rate. So we know the scatter light in a perfect integrating sphere is same everywhere. Doesn't matter where your light is calculating. And so we actually use a constant ratio, and over time that's a constant. In some cases, we actually see reverse. The measured light is actually lower than the calculation. And we actually have to look back into our data, and we found out this particular case, posterior mediastinum, the light is blocked by the tissue because of contour, and our calculation didn't take that into account. It just calculated based on the distance between the treatment area and light. But we are not as sophisticated to know the contour. Actually, the other contour is blocking the light to the area of treatment. So that actually is one area we're still trying to work on. But anyway, the important thing for us really is to ensure the calculation and the measurement match with each other. Then we can more or less believe in the calculated light fluence and other side, which is the purpose of this exercise, so-called navigation-based guidance. We want to actually use that to guide the surgeons to decide where to put the probe into. So we actually developed a sort of a correction method, which is to that type of correction. Every 30 seconds, we recalculate the calculation by comparing the calculated value with the measurement. We measure the instantaneous fluence rate. If the difference, we call that correction factor, is more than 5%, we apply the correction. If the difference is less than 5%, we let it go, doesn't apply for correction. And then we find we actually have to do a second type of cumulative corrections. So the second correction is actually a little longer time than the first times. First, the 30-second correction, which is 150 seconds. Every 150 seconds, we look at the cumulative fluence up to that times. Here, where every 30 seconds, we look at within that 30 seconds. Here, we actually look at the cumulative over the entire treatment up to that times. We match them under that condition. And this is actually applied to, the data I'm going to show you is just use direct light calculation, okay? So you can see, at least in this case, they're jumping around a lot, but you can see in the end, all sites, you can have the calculation, which is these dots, agree with the measurement, okay, in all sites. And that will give us some confidence in just using this correction method. We just force them to, I mean, the correction, one thing that's a little tricky is the correction actually has to be same correction. You have to correct all the sites, 7 sites simultaneously. So there is some truth in, the model itself to match the data between the calculation and measurement. And here is, without correction, what we have, some of the result, the total fluence distribution. What we do is we take the pleural cavity, which is actually can be determined from a navigation system. We unwrap it into this 2D against the angle, so-called horizontal angle. So we basically unwrap it by, 360 degree. We unwrap it onto a surface, and the vertical direction is in this direction, up and down direction, okay? And to the surgeons, the most important thing is they want to make sure the surface is same color. That's all they need to know. They make sure the surface is same color, and then they get the uniform treatment. And you also know where the detector is. So this marks where the detector is. And that's, in existing treatment, we know where the detector is getting the light, right? You can see this one is extremely homogeneous, but wait, this is before corrections. So we know the calculation doesn't really agree with the measurement. So really the calculation is not that correct. Here is the one that with the correction applied to it. And you can see actually the current method is not too bad. This is the ultimate treatment after, over 2,000 seconds of treatment over the whole course of treatment. And they are reasonably good, except for some point there's some hot spots. So this is basically indicative, give us a tool to decide how we can ensure the light distribution, and potentially, because it can be done in real time, you can have the surgeon use this method to paint the surface, basically, and become a painting method. Some summary of the comparison for here is 7 patients. We have used direct light only. If you don't do corrections, you don't have any agreement. Here is direct light only with the correction and without taking into the scattering light. We only have 1 patient that we can get reasonable agreement. The other patient, we have majority of them cannot agree. I mean, this is the maximum difference from one of the 7 detectors, so they have to be compared simultaneously. This is very encouraging if we take into account the scattering, which actually in our case is just a constant. We introduce a constant factor, which you can decide from this correction mechanism. You can have majority patient agree. One does. I mean, I am using close to 15% as agreement. So the majority of them can actually have a reasonable agreement, which will be very good for us. So we talk about light, and we certainly currently has lacking a good way of determining the sensitizer distribution. We only can know the sensitizer at a few different points on the surface. We want to use this method to determine the PDT dose. But in the second part, really the important thing is because we know the PDT is oxygen-mediated effect, you really have to develop a model based on single oxygen to take into account the single oxygen consumption, especially for this kind of treatment where your light fluence rate is changing from 0 to 400. So you have a really inherited light fluence rate change, which the PDT model will not take that into account. At the current way, Brian's single detector is the ultimate. If we can have a way of detecting it directly in clinic, it is the best we can. But at this moment, the way we can do really is using the existing explicit dosimetry, try to develop some model which will allow us to use it in a clinic setting to get single oxygen directly. So this is the same so-called Jabrowski diagram that you have seen again and again in the previous lectures. So you basically have a sensitizer excited into the excited state. Some of the sensitizer get onto this treat-bridge state, and they will interact with the oxygen to generate a single oxygen. And this is the quantity that interact with the either the target cells to do the damage, the treatment, or photobleachings. And Brian has mentioned about using photobleaching during the treatment, and that could be one method. But what we are currently doing is basically based on explicit dosimetry of individual quantity and try to tie them up using this so-called macroscopic model. The complication of using this model is, you know, all these parameters are known. All this cross times is, you know, very little literatures. There is some existence in the cell or in vivo studies. So first step is to determine all these parameters before one can use it in clinic. So we actually have tried to use a mouse model to determine this quantity. And as Brian has mentioned before, those parameter is sensitizer specific. So for specific sensitizer, for specific tumor model, we have to decide a specific parameterization then before we can use it. So it turns out that one doesn't have to determine all these 7 cross-section lifetimes directly. You actually can determine by combining some of these parameters. In here, we have identified 4 parameters: sigma, psi, G, beta. So 1, 2, 3, 4. If we can determine these 4 parameters, and these parameter is a ratio of interaction times, okay? And I'm not getting into details. So we just have to determine these 4 parameters. And we are currently doing some mouse studies using a linear source interstitially, which will allow us to determine the necrosis depths in a mouse model. This study actually is done photofilm. We started with photofilm. We actually is going into HPPH2. We have some results presented recently SPIE this last year. But the necrosis depths for various treatment conditions can be used. So we change the fluence rate from very low to very high, and change the total treatment time, you know, from very short to very long. And then we quantify how much necrosis you get. And that will allow us to do a fit to determine these parameters. This is actually one of the latest data for photofilm at least. You can determine the sigma, these 4 parameters, as well as the single oxygen threshold dose, because that is also another important quantity to determine how to use it in clinic. Now our results varies a little bit. I think this is the one that's published initially, where we use the published literature data for one of the sigma. These are some of the parameter is in existence in Tom Force's lab especially. And of course, Mike Patterson and Brian Wilson has done a lot of study too in different tumor models. So for example, beta factors is quite well known, and we kept it constant. Here, actually, these data actually is obtained by doing a complete inverse, solving the inverse problems, and let all the parameters flow. So this is our latest for photofilm, at least, using a rift tumor model for rift tumor treatment. So what's the use of it once you get that? Well, one thing actually is intriguing is this. If you look at the distribution of single oxygen, one can actually calculate the distribution of the so-called quantity reactive single oxygen. This is a calculation of the reactive singulation for point source, I think, as a function of distance from the point source. You find that they actually have a very sharp edge. I mean, this is actually protein log scale. So this edge is really sharp. And that's interesting because you find in PDT treatment that you have a very sharp necrosis edge in the treatment. And that seems to be using singlet model, you can sort of correlate the necrosis depths with this edge, which is not predictable by the light fluence rate itself. So light fluence rate is changing, a lot. It doesn't say there's any specific edge. And here with single oxygen, you can find a edge. And that doesn't change too much. If you change the treatment times, change somewhat. So that is potentially can be used for the modeling. So the goal is really using this so-called navigation system to get the light distribution for our pleural treatment. And we are still working on how to determine this two-dimensional distribution of sensitizers. But one good thing is we actually found out the sensitizer distribution before and after our treatment, at least all the treatment I've seen, photodynamic or HPPH, they don't change before and after. So one potentially can determine this distribution in one measurement before PDT, and assuming it doesn't change through the treatment, then one can actually determine this so-called reactive single oxygen by these simple formulas, which is a function of PDT dose. Okay, this is a product of sensitized concentration and the light fluency over time, modulated by the single oxygen quantum yield, and this quantity is the one that forces us to do calculation, because the oxygenation itself is the quantity. We cannot determine that using any existing method. So we actually have to calculate the oxygen as a function of time. And that quantity, if you put it in, then you can determine this single oxygen. The only modulation of this additional single oxygen-dependent single oxygen quantum yield is taking into account the light fluency effect. So really, is the first step for us is to determine the PDT dose in clinic settings, and the next step is to take into account the additional light fluency effect. So this quantity really can be relative, of course, as a dosimetry quantity. And the good thing is that allow us this model, and also there's a lot of breakthrough in the preclinical studies, which will allow us to tie up all these quantities, oxygenations, light fluency, and drug concentration all tied up together into a quantity, hopefully, which will correlate directly with the outcome, which is our hope, of course. So in conclusion, we have this explicit PDT dosimetry that can be used to determine the tissue optic properties, sensitized drug concentration, oxygenations, which is actually hemoglobin concentration, and then which actually can be used to use our model to correlate to the oxygen concentration in tissue. And in vivo study strongly suggests heterogeneity of optic property and sensitized concentration require individualized determination of these quantities. At least one way using navigation system, which will allow us to deliver uniform light distribution. Some of the model method which will complement this technique and tie up all these quantities together. Okay. In the end, I want to acknowledge all this big group effect. This is the physics group we have here currently. We have a large number of physicians working on these cases, statisticians, nurse practitioners, and biologists doing more fundamental research. Thank you very much.
Thank you. For that animation that the light source was moving around,
Yeah.
I wonder why you didn't use a different kind of geometry. I mean, that have a source and the detector at the same fiber compact together.
We actually are looking into that too. This is, this technique actually is developed over the last 20 years. Back in the late 80s, they used this technique for intraperitoneal treatment and for pleural treatment. It was established at NIH back in 80s. And the advantage of this method is you sort of, can deliver the light to wherever you want to go, because all you need to do is move the light source to wherever you want to move to. The disadvantage, as you probably are pointing out, you are not ensuring the uniform light. We actually cannot use fixed light source geometry with a point source, because we know that it will not be uniform, right? Because the contour geometry. And the better method probably is using some kind of blanket. We are doing some development of flexible source. So you have this fabulous LED light source, and that will be a great method. And all you need to do is make it flexible. So you can just put it in an area that you want to treat. And yeah, so this is one area we are looking into to develop some more flexible blanket to treat. And we probably are thinking about initially to this Pleural is a good area to use. And then you can use that easily for boosting, for example, boosting some areas. Just put a blanket there. And I'm sure the light fluence rate uniformity will be improved drastically with these other alternative methods, yeah.
Another question. About that constant value, do you need, are you able to predict this value, or you need to run a protocol every time before each treatment?
The volume we cannot predict. We basically use, that's the reason we use in vivo light dosimetry. Then because of we use in vivo light dosimetry, all the effects, the volume, the size, the shape, the optic property is all taken into account by this one detector that will give you the true light fluence rate at that point. And we accumulate over time. So we can determine that light fluence rate correctly. We know that because of these detectors. And now with this system, we kind of also know sort of the distribution on the entire surface. So that is the way we do. Our plan is everything is done in real time, in the OR setting, and all these quantities determined in real time. We probably has to do a lot more work because, as you can see, we are only determining the pleural cavity. But when you're actually doing the lung treatment, you are treating tons of critical organs. The heart is getting treated. I didn't mention the heart at all. We have one detector on the heart. And we actually have to do a lot of study to register our surface with the CTs, and potentially you can do a lot more. But there is a lot of work to be done.
Yeah, exactly. It's my question. I just want to know, over 1,000 treatment, is it possible to, for example, just extract average value of this constant value? And
we haven't done that because it's patient-to-patient dependent, as I said. We have tried to do that for a systemic, to a patient I've shown before. You have a mean value. But that mean value, it's giving you a good indication that if you know the size, you pretty much know how long you want to treat. That's all you need to know. But it doesn't preclude some patient has a very weird contour that you have to treat longer or shorter time. So we still are dependent on our in vivo light dosimetry to decide how long we actually need to treat. We have some secondary way of estimate how long it will be, but it's that's the estimate.
We know that the photosensitizer distribution is very non-uniform.
Yes.
And is three-dimensionally distributed.
Yes.
And that somehow affects the production of singlet of oxygen.
That is what
which I think locally will be the best dosimetry. But it's very hard to do. And we have fluctuations on the concentration in a very thin layer, right? From one position to the next. So the question is, how really valid is a process that does not cover all the space? I mean, or in other words, how the heterogeneity, non-uniformity
yeah.
of concentration of photosensitizer compromises a real dosimetry?
A very, very good question. This is a question that make people hesitate of relying too much into this kind of model. So I actually, so what we are doing now is we try to build a model. This is an empirical model in a sense too, because it derived the parameter which most likely is not directly correlated to the cellular level interaction times. But it will give you a parameter which will get the best fitting to the necrosis depths, okay, empirically. And then we got those parameters, we can do the calculation, and I mean the first question also is you have to demonstrate the calculation is feasible to be done in real time, which I think is very feasible now with our current computational power of computers. We actually sort of using this methodology because we know this return back to the PDT dose. So PDT doses has been a valid concept, have been used for over a long period of time, a product of light and drug and you want to make sure that is given a proper dose. So we basically just slightly modulate that with the sigma eta values. And the hope is really this, this modification is not that drastic, which our hope is really to improve the dosimetry, take into account the fluence rate effect. I mean, you're right that I have been relying on, for example, calculation to get oxygen depletion over time, and that calculation may or may not be right. But really our ultimate goal really is only to look into whether the fluence rate effect, which could be large because you are changing from 0 to 400 milliwatt in a period of time, that's changing back and forth drastically, which is not taken into account, and how this can be taken into account in a sense. So I don't know if I answer your question. I guess my answer is we don't know, actually, in the long term. What we're really trying to do is just to build this model one step at a time to get some empirical quantity. I mean, the reactive single oxygen here really is an empirical quantity anyway, calculated quantity. That could be related to this, this quantity.
Actually, can I comment on this general issue? I mean, I think one of the realities is that we have to live with reality. Right. So the reality is that the light distribution is not uniform, the sensitizer distribution is not uniform, the oxygen distribution is not uniform.
Yes.
And there's nothing you can do about it.
There's nothing I can do about it.
So the question is, how do you manage that uncertainty? Right, so it's not a matter of eliminating the uncertainty, it's a matter of managing the uncertainty. And so one of the ways that we've been thinking about it for the prostate is, based on cumulative experience, you find out certainly the average of these quantities, but you find out the variance. So you find out the range of variability from patient to patient. And then you say, okay, when I go into the next patient, that is my uncertainty.
Yes.
Now you design the treatment that encompasses that uncertainty. And you say, if we are at this limit in terms of the product of the oxygen, light, and sensitizer, then we have a certain risk of undertreating. If we're at the other limit, then we have a certain risk of overtreating. And now you make a clinical balance that says which of those is the more important to give weight to. So I think we must, I think in PDT in general, until we have some fantastic dosimetry that really allows us to make all the measurements we need dynamically and feedback, I think we need to start thinking about how we give the treatments living with that uncertainty.
I agree, yeah. I absolutely agree with that.
I mean, most of the dosimetry being established here is based on the total singlet oxygen production. We know that most of the times the singlet oxygen production rate is even more important than the total production afterwards. I mean, like, according to photochemistry laws, you can assume that if you deliver the same energy, you're going to make the same reaction. If you deliver 2 joules, you're going to have that amount of singlet oxygen. Don't you think that we might be heading toward the wrong direction of establishing a really consistent dosimetry?
Well, I think
regarding the rate of the productions and not regarding the other ROS productions. As well as the
I
oxygen.
want to clarify one thing, okay? So really, I don't know if I probably didn't make it very clear. So if you look at this diagrams here, this quantity we have calculated here is really not the single oxygen production rate.
Yeah.
This quantity really is the single oxygen reacted with the tissue targets. And that is the reason this quantity becomes cumulative quantity. So as you're treating longer and longer times, this is a cumulative reactive single oxygen. We are using the unit as concentration, so one kind of saw that is instantaneous concentration, but it's actually is accumulation of sensitizers over treatment time is what we are quantifying. So this is so-called reactive synchronous oxygen really is not instantaneous oxygenation at the time, at any single moment. So as a result of that, as you can see, the resulting quantity we're getting really is the integration over times. Are you saying this is not, you don't like this particular method?
I mean, because we have some data that we fix the dose that we are delivering, we fix the fluence that we are delivering, and we vary the fluence rate, and we get so much different results after it.
Yeah, that is true.
So if we depend on tissue optics, we're going down and down, we're going lower and lower fluence rates.
I mean in the mouse study, you can have a controlled environment where you can you can have a constant single oxygen concentration over the entire treatment time, which is not changing at all. But and that you can correlate the condition, each conditions with the necrosis result. That's what we actually did. We correlate each conditions where the single oxygen production is constant during that each conditions. But then when we try to apply it in a clinic settings, you cannot maintain a constant single oxygen productions. And we have to do the other part to do a dosimetry, because just impossible to maintain a constant production conditions. That's just not possible in our case. In this kind of cases, maybe in the skin treatment you can maintain a particular distance or life fluence rate, but in most of general PD treatment, one really cannot maintain a particular condition for single oxygen. And this is kind of attempt. Once they establish this, so really the effect that you have mentioned before, different single oxygen production rate has different outcome, which means necrosis, is the parameter we actually build into these parameters. Those effects we take into account by building our set of parameters to match the results.
That's great.
And once we have determined this set of parameters, we can recover whatever you have seen in the mouse condition, for example, where you have a controlled environment for each particular single oxygen productions. We just expanded basically in the clinic setting to a condition where you have no control of how much single oxygen is produced. But our hypothesis really is the cumulative reactive single oxygen is the quantity that determines the ultimate outcome.
Yeah.
And that's also the same hypothesis we use in our mouse study.
Okay. What about the other ROS formed?
That is a very good question. Actually, if you want to really go deep into the mechanism, I didn't really talk too much into it, but the single oxygen threshold dose we are getting here, 0.5 to up to 1 millimolar, is about a factor of 10, less than what you actually determine from cell studies, where you have to have that level. So in reality, our model is completely empirical model that may be including some other vascular effect or immunological effect, which is making the PDT much more effective in killing. So really, our effect, I would say, is an empirical model that it's based on the singular oxygen production, but in the end, it includes all the effects, including the vascular effect and maybe your so-called IOS. But I cannot defend this model will be able to model these other effects, of course. But we just do the best we can, and hopefully, the condition in our mouse study is close enough to our clinic settings. So really, the best disease site to investigate is, like Brian said, is the prostate site. Those are much easier site where you can have a more well-controlled light fluence rate, because you can put the light to wherever you want to. That actually is a site I'm really looking forward to. And then the pleural, this cavity, really is a very, very complex case. This is where you don't even know how you calculate light, because it's right in the interface between the scatter medium and non-scatter medium, with lots of blood inside and the arbitrary cavities. It's as hard as you can imagine. So everything is in here. So this probably is what we you know, we have to, you know,
Yeah, we need to
develop
keep
over
getting
over
closer
a
to
period
it.
of time, a long period of time that you can ultimately, you know, solve that.
Okay, thank you.